Hey, good morning, everybody. Welcome to the last week of classes. So just to put where we are into context, we're going to spend the final 3 lectures on ion channels and pumps today, membrane transporters on Wednesday, and then we're going to, armed with that biology and understanding, go into the pharmacology that affect both of these types of entities within cells. A little note on the weather. I've been watching Wednesday. There's some chance of a snowstorm, and then sometimes they say there's not. But in the unlikely event that we did get snowed out on Wednesday, I would record the lecture and post it the same day. I know there's like a university makeup day, like I think it's a Saturday before finals week. I wouldn't do that to you. So I would, in the event of snow, I'll record a lecture and then post it, okay? But anyway, this is a really interesting final series of topics. You'll see this also in greater detail, additional detail in the neurobiology module, the cardio module, and even in the renal module, and even oncology because of cancer resistance. So these, ion channels and membrane transporter proteins are very important in a lot of processes, and I think that will become apparent. So a little bit of background to get into this. Ions and ion channels, which allow ions to go in and out of the cell, are important for some very highly significant processes. They control the pace and the pumping action of the heart, for instance. So obviously that's quite important. They regulate secretion of hormones. But then also very important, they generate, they're involved in the generation and transmission of impulses, nerve impulses in the brain and the peripheral nervous system. So, important for the heart, important for regulating the nervous system, so very important functions. And of course, you get all sorts of problems when ion balances are not managed properly in the body. Cardiac disease, neurobiological dysfunction, they're just important in so much stuff. So this is kind of an older figure, but it still pretty much illustrates what I want to note. It just looks at the world of targets for drugs, and of course there's GPCRs. But I draw your attention to this part of the pie chart here in purple, and this part here. These are basically targets that are ion channels, ligand-gated and voltage-gated ion channels. So even though that together comprises maybe only 15% of drug targets, it turns out that the physiological processes that are regulated by ion channels is so important that a huge amount of pharmacology has been developed to help address this. So, a little bit of general stuff about ion channels, and then we'll get into some of the details. So as the name implies, ion channels are like pores. They have a central pore that allows ions to flow into and outside of the cell. And they typically have a whole bunch of transmembrane domains. Sometimes they're single proteins, other times they're groups of proteins that come together to form a single channel. And they can sense all kinds of things. They can sense voltage gradients across membranes, for instance, which is in our system is usually a difference in membrane potential mediated by an imbalance in the amount of charged ions outside of the cell versus inside of the cell. They can also, and they can open and close in response to things like voltage. They can also sense, in some cases, chemical and even mechanical signals, and those signals can cause them to open and close. So, when they're open, ions can freely flow across the cell membrane. Okay, and it's very important in regulating electrical signals. So there are many types of ion channels, and here is a diagram that kind of summarizes some of them. What you can see here is this yellow bar basically denotes the plasma membrane, and the exterior is on top and the cytoplasm is on the bottom. And you can see that there are certain channels that are always open. For instance, certain potassium channels are always open. And that allows potassium ions to freely diffuse across that membrane. Now, notably, that's only going to happen when there are more potassium ions on the inside versus the outside. And then, given time, these ion concentrations, exterior and interior, will come to an equilibrium, and there will be no charge difference across the membrane. But there are other ion channels, in this case a sodium channel, that are voltage gated. And what happens here is that when the voltage, I don't know why this is advancing, but when the voltage reaches a certain potential, the ion channel itself can sense that voltage, and the sodium, the pore will open, and then sodium can then move across that channel. So it's actually a voltage differential that is changing the ability of that ion channel to open. And we'll talk more about how that works. The next case I want you to know about are ligand-gated channels. So these are channels that are ordinarily closed, but when a ligand binds to that channel, it causes the channel to open, and then ions can flow through the channel, okay? So this one's triggered by voltage differences. This one is triggered by ligand binding. There are also channels that can open and close in response to intracellular signals. And of course, that would have to impact the cytoplasmic side of the channel, okay? And these are basically some of the major classes. So channels that are always open, allowing for passive diffusion of ions, voltage-gated channels, where a voltage difference across a membrane triggers their opening, and ligand-gated channels, where ligand triggers their opening, and some that even sense signals from inside the cell. And some of these interior signals include things like signals that are originally started by G proteins. Calcium, there are a lot of cases where calcium can actually open channels for other ions to flow through. So they are pore forming. They are usually multimeric, meaning that more than one protein species gets together to form the complete channel. And they open and close in response to chemical, temperature, mechanical signals, voltage. And once they're open, basically ions will rush into or out of the cell, and that will happen until that ion reaches equilibrium on both sides, okay? Unless something else changes it. And interestingly enough, it's this phenomena that propagates action potentials and nerve signals in neurons. Okay. So you can further classify ion channels in the following way. And some of them are active transporters. Some of them are called secondary active transporters, co-transporters, exchangers. And here's how this works. So if you look at this on the left side here, we're looking at 4 different ions. Ions 1 and 3 are at high concentration outside of the cells compared to inside. Ions 2 and 4 are at high concentration inside the cell as compared to outside. Okay? So an active transporter involves energy. Okay? So here is ion 2. It's at a lower concentration outside than inside, but you can still get these ions to move across against the gradient because energy is being used. Typically that's ATP. In this case, ion 2 is being imported into the cell against a gradient, and ion 1 is being transported outside of the cell, again, against a gradient. Of course, to do that, you have to burn a lot of energy in the form of ATP hydrolysis. Then there are also just ion channels that are completely passive. They're always open. So here's ion 1, high outside, low inside. And ion 1 will just freely flow across this open channel, open at all times, until it reaches an equilibrium. Now, here is another example, a co-transporter. So ions 2 and 3, one is high and one is low, but you can get them both to move across because sometimes the energy mediated from going from high to low, ion 2 for instance, can allow the co-transport of another ion against a gradient. Okay, that's kind of an unusual case. And then there are so-called exchanger channels in which one ion can move out, one ion can move in, always with basically, and so, you know, it can exchange. So there are many channels that transport single ions, and you also see a lot of examples of co-transporters or ions that or channels that exchange two different ions through the same channel. But one rule of thumb is that the passive channels always have to transport from high to low gradient, whereas the active channels can actually move ions across a gradient, against a gradient. So there is a very important channels that actually transport sodium and potassium across membranes, and they can do so in many ways. So let's start to now think about how channels work together when you have more than one in the membrane, which is what's really going on. So on the left is a passive potassium channel, and it will, and you can see that there's more potassium is on the inside of the cell than the outside, potassium ions. So this potassium channel will allow potassium positively charged ions to efflux the cell just passively. Here is an open sodium channel, and the sodium channel allows sodium ions to come into the cell, again, along with the gradient, high concentration here to low concentration, so that means that the sodium will flow in. So if you just had these two channels, the green ones on the left and the right, what would happen is that sodium and potassium would eventually come to an equilibrium across this membrane, right? It'd be the same concentration of sodium and potassium on the outside as on the inside. So to actually set up a gradient, you have things like this pump, which is actually forcing sodium ions out and potassium ions in, again, with the use of energy. And basically, when its energy is used, oftentimes we'll call these pumps. So what this is doing is setting up a charge differential across this membrane, even though there's leaking going on with the passive diffusers channels, okay? So it's kind of like an analogy, be like you're in a rowboat out in a lake and you pull the plug in the bottom of the rowboat and water is coming in, but you're also bailing, okay? So you could bail out water, which is an active thing. It takes energy, right? And even though there's water leaking into your boat because the plug is pulled out, you can still keep the water out of the boat by expending energy. And that's kind of how these things work. But these exchanger pumps are what are involved in maintaining a resting potential across cell membranes, and that's very important for ions, or excuse me, for neurons. Okay, so let's next get into voltage-gated ion channels, and these are kind of interesting. So here is a voltage-gated kind of generic ion channel, in this case, a channel for potassium. And what you can see is that you can actually measure a voltage across the membrane. basically because you have more potassium on the inside than the outside. Okay? And this channel is closed, but if for any reason that concentration difference between potassium ions changes, and you go from minus 70 millivolts to minus 50 millivolts, that triggers the channel to open, and then potassium will rush across the membrane until equilibrium happens. And that is a phenomenon called, this is a polarized state, and this is a depolarized state. So it's called depolarization. So when a nerve cell fires and transmits an action potential, we call that depolarization. That's what triggers the actual change. So let's look then a little bit more detail about how these voltage-gated ion channels work. So again, this is the outside of the cell, this is the inside of the cell, and these voltage-gated channels can exist basically in 3 conformations. And by the way, the pore in a channel is very selective. So, you know, if this is a potassium ion channel, only potassium can fit through the pore. If it's calcium, it would be a different channel, and the pore size is different. So these channels are very selective for the particular ions that can go through them as well because of features of the pore. But anyway, we have this situation where there is a resting state, and then we have a second configuration where it's open and ions can move through the channel, and then a third period called refractory period, which is interesting. So anyway, within the structure of the proteins, actually involved in alpha helices and certain extra membrane portions, these channels have voltage sensors. And so these are charged amino acids that can respond to the voltage differential. And what happens is, here it's closed, but when the voltage changes enough, those voltage sensor portions of the protein change conformation, and that opens the pore, and sodium can flow in and through the channel. But interestingly enough, after enough sodium has flowed through, this channel can adopt another configuration where the actual pore is still open, but there's like a little plug, which is part of the protein that can actually plug it up. And this is called the refractory period. So this is a channel that is otherwise open, but this plug prevents ions from flowing across. And so it will cycle between these 3 states when, for instance, an action potential is transiting a neuron. And during this refractory phase, it's an inhibited state, and basically once the voltage goes back to this resting potential, that little plug will unplug, and you'll be back ready at this state. So this is kind of like a prime state. Here is active channel activity, and then there's a refractory period where the ion gradients are being reassembled, and then the cell can respond again. So these voltage-gated ion channels are interesting. Here it is closed, and here it is open, and you can see that some of these alpha helices actually have embedded charges in them that can sense voltage potentials. It's, you know, voltage is just a charge differential, and when that voltage changes, it can cause a conformational change, and then ions can flow through. So it's a pretty cool protein because it can actually sense voltage differences across a membrane. And it's because there are charged amino acid residues in these alpha helices. So this gets very complex. We're going to kind of get into a little bit of neurobiology. And so here is a diagram of a synapse. You have a presynaptic neuron, the synapse itself shown here, and then this and then the postsynaptic neuron here. And you can see that there are voltage-gated channels up here, shown here, one ligand-gated channel. But at any rate, what's going on is I want you to see that there are all these different channels. But what these channels in the presynaptic neuron are actually doing is eventually they're triggering the ability to release neurotransmitter. And these channels down here in the postsynaptic neuron are going to ultimately respond to ligand. And here we have the neurotransmitter glutamate, which can trigger this channel to become active. And you have other channels that are just maintaining gradients and things like this. So kind of a complex situation. Okay. If you look at, and we'll just quickly run through a couple of these things just so you can get a general feel for the structure of these channels. Here's a voltage-gated potassium channel. It only has 6 transmembrane domains in the protein, but 4 of these proteins get together to form the complete channel. So this is a homotetramer, okay? There's a pore in the middle, and some of these helices participate in voltage sensing, and then there are others on the inside that are modulating intracellular domain, intracellular signals. So a lot going on. Here's a voltage-gated sodium channel, and this one is one single protein instead of 4, but it still has a total of, 4 domains. So this one is 4 different proteins that assemble to make the complete channel. In the case of the sodium channel, all 4 of these domains are in the same protein with big loops, cytoplasmic and extracellular, but it still has the same thing. The green ones, I believe, no, some of these colored ones are the voltage sensing helices, okay? Yeah, so similar. Here's a calcium channel. Again, this one is one big protein with 4 distinct domains that just kind of loops together and forms a complete channel. And this one can be ligand activated in addition to voltage sensing. So I guess the take-home message from all this, going through these different channels is that they tend to have kind of a tetrameric structure, but sometimes it's 4 separate proteins that get together, and sometimes all of those individual units are contained in the same protein, but it can fold into a channel. Okay. So let's see now how this all works and talk about action potentials. And of course, action potentials work in neurons, right? And so this is a diagram of a generic neuron. You have the cell body, the nucleus is there, the DNA is there, proteins are made here. And then you have the long axon. This is the part of the cell that a nerve impulse will transmit through, which is called an action potential. And then at the very end, you have these, the nerve cell makes connections with other neurons. And so this is where all the synapses are. And this axon can actually be very long, sometimes a meter or so long if you have a single nerve cell going through like a limb, which happens. And sometimes they're short, but the axons really are long and they make all the interconnections between neurons possible. Another thing that's interesting is that this axon in neurons is coated with myelin sheaths. And so this is done by Schwann cells that lay down myelin. And this is like an insulator. So there's no ion flow possible where a myelin sheath is present. But there are, and Schwann cells are specialized cells that actually lay down this myelin. It just kind of loops around the axon. But in between, the myelin sheath is interrupted by a very important region called the node of Ranvier. And these are places where there is no myelinization, and that is where ions can flow across. And so this has the effect of increasing neurotransmission, increasing the speed and distance that a nerve impulse can travel. And so to tell you a little bit about channels, what's going to happen is this neuron starts to decides that it's going to depolarize and transmit an action potential, and you'll get depolarization there, and that triggers depolarization at the second node, and then the third node, and then the fourth, and it travels on down the axon, kind of pop, pop, pop. And this really keeps things coordinated and allows the action potential to be propagated over a very long distance. And so depolarization at each node triggers the next one down the line. Okay, so let's now focus in on that depolarization in a little more detail. So what's really happening here is something interesting. The cell can exist in a couple of states. Normally it's at the resting state, and there's a resting potential where there is a kind of given charge difference between the outside of the cell and the inside of the cell. In the case of neurons, sodium and potassium are especially important. So in a resting neuron, sodium is high on the outside of the cell, and potassium is at a high concentration on the inside of the cell. But when this nerve cell is triggered to fire, an action potential is initiated, and what happens is that sodium rushes in, okay, and that's depolarization. And then during the next phase, repolarization, and actually then potassium can run out, okay? And then this nerve cell has then fired, and you'll notice that these are in a state of disequilibrium during the resting state. Then depolarization happens and ions are moving. And then after the nerve signal has been transmitted, you enter a resting state, and you have to regenerate those ion gradients, and that's done by a sodium-potassium exchanger pump, and that takes energy. So while this is going on, the nerve can't fire again. It's kind of a one-time deal. An action potential occurs, ions flow across, and then the nerve cell can't fire again because it has to take time to regenerate that ion gradients. Okay. So this exchanger pump is very important, and it's called the sodium-potassium exchanger pump, and it takes energy. And so this is an interesting thing. It will pump sodium across in one direction and bring in potassium in another direction, but it takes energy. And here is how it works. It's kind of, so there's a cytosol, it has these two different configurations. There's a binding site in this pocket in the pump that can accommodate 3 sodium ions, and then ATP is used to change the configuration of the pump, and so now these 3 sodium ions can be ejected, but that opens up a place where 2 potassium ions can fit in. And then phosphate is released and kind of pops back into its original configuration, and the potassium is released into the cell. So it's always 3 sodiums going out, 2 potassiums coming in, and it's an energy, the first step of this is energy dependent. So kind of a cool little mechanism. But this is the pump that actually generates the electrochemical gradient across the membrane of a nerve cell. Okay? And without that pump, you couldn't recharge the neuron back to its resting potential. Okay. So, how does this work? Well, neurons actively transport ions across the membrane using ATP. This is just kind of a diagram to go through these steps in a little more detail. 3 sodiums will come in, 2 potassiums go out. So this sodium-potassium exchanger pump is doing this. And what you'll see is that every time that pump works, 3 sodiums go out, 2 potassiums come in, and that's going to generate a membrane potential difference between sodium and potassium on the outside of the cell versus the inside of the cell. And this is actually working against passive diffusion channels that always, and they're called leak channels in neurons. So there's a leak channel for potassium, another leak channel for sodium. So if you didn't have this exchanger pump, everything would come to equilibrium. There'd be no such thing as brain activity. And so this sodium-potassium exchanger pump is always working hard using ATP to generate these gradient differences. Incidentally, your brain uses a heck of a lot of energy. Even though it weighs like 4% in a human, it uses 20% of your energy. And so a lot of that energy is making membrane potential across your neurons. It takes a lot of energy to do that. And glucose is used, which then makes ATP through a lot of biochemistry. But that's why the brain is a very kind of energy-heavy organ. In fact, it uses the most energy per unit mass of any organ in the body. Okay, so here is a cell that is now ready to transmit an action potential thanks to the sodium-potassium exchanger pump. More sodium on the outside, more potassium ions on the inside. Both of them are positively charged. And what happens is, there's a big membrane potential, so it's positive on the outside of the cell. And basically, there's an electrochemical gradient for sodium and potassium, but they're in opposite directions, okay? And the leak channels will degrade that if it weren't for the action of the pump. Okay, so primed neuron, ready to fire, has a bunch of sodium on the outside and a bunch of potassium on the inside. And so if you follow an action potential and look at what a cell experiences that's just going through an action potential, you'll see that it's resting at 70 millivolts, but when it reaches minus 55, then depolarization happens, and it goes all the way up to plus 40, and this is when the action potential is being propagated, and then it has to repolarize, and even after that, there's a refractory period where no action potential is possible, and then it gets back to the resting state, and then it's ready to fire again. So you have these kind of sequential steps: resting state, depolarization, repolarization, a refractory period, and then you're back to the resting state, because you can actually measure the voltage potential across neurons with things like patch clamping, for instance. So the depolarization is pretty cool, and it's triggered by a voltage-gated sodium channel. This is neuronal firing. So here's what happens in the depolarization phase. Basically, sodium's gonna go out. Sodium's gonna come in, and that basically is what's doing the action potential. Okay. But after the action potential actually happens, the voltage-gated sodium channels inactivate, and then the potassium channels open, okay? And potassium goes out, okay? And so basically it's first sodium and then potassium. And then the refractory period here doesn't have much voltage difference across, and basically the sodium-potassium exchanger pump is then going to reestablish this gradient difference between potassium and sodium so it can happen again. So I think it's good to kind of have a feel for that and just study this a bit. Know that minus 70 is the resting state, and depolarization happens, sodium ions are coming in, and then during repolarization, potassium is coming out, and then you have a refractory period and a resting state, basically. Okay. So, physiological functions here of all of this. Sodium is a big extracellular cation, and we'll get away from neurobiology a little bit. It turns out that sodium is really important for osmotic pressure gradients between the inside of cells and the outside of cells, okay? And so that's going to be important for all of your cell types. It's really important in the vasculature. And so sodium tends to be extracellular for the most part. Potassium is a big-time intracellular cation. And then, like I just mentioned, it's involved in making the resting potential inside neurons. It's also very important for muscle fibers because muscle fibers do this as well. And chloride is a big extracellular anion, also contributing to osmotic pressure. Okay. So already you can see that, you know, there are some very important things going on here. Important for neurological function, important for maintaining osmotic balance in the cell. It's also important for cardiac function. Even interestingly enough, channels are also important for pumping hydrogen ions into the stomach to make stomach acid, for instance. And those are active pumps. Okay, so those are kind of the basics of all of that. Let's next just spend a little bit more time getting into ligand-gated channels. Okay, so ligand-gated channels are really quite different. Before, you know, with a voltage-gated channel, it's really a voltage difference that is opening and closing the channel. But in the case of ligand-gated channels, it's a ligand of some kind that binds to this channel itself, usually on the exterior side, on the extracellular side, and that will cause the channel to open. Okay, so for instance, here is a channel, actually a sodium channel, and it's closed, and there's a, you know, there's a lot of sodium on the outside versus the inside. So there's the potential then, if this channel opens, for sodium to enter the cell because it's going to try to destroy that disequilibrium, right? It'll try to reach equilibrium once it's open. But in this case, the ligand is acetylcholine, and now that's a neurotransmitter. So acetylcholine comes along and associates with this channel, and that allows the channel to open. So these ligand-gated channels are especially important, for instance, in the postsynaptic neurons, because that's where the receptors are for neurotransmitters, okay? So a neurotransmitter channel, you know, ion balance actually in the presynaptic neuron is going to be involved in allowing neurotransmitter release into the synapse because it regulates the secretion of neurotransmitters in the synapse. But once that neurotransmitter is in a synapse, it can then bind to a ligand-gated channel in the postsynaptic neuron, and then ions can move across that channel. And since you're changing the charge difference in the postsynaptic neuron, you can trigger depolarization, neurotransmission, and an action potential can be initiated. So it's kind of a combination of work between ligands and channel opening and closing. Okay, and incidentally, calcium can actually sometimes affect channel opening for other ions. So these ligand-gated channels are interesting, and I guess the key here is that ligand-gated ion channels have to have two binding sites. There has to be a binding site for the ligand itself, and usually on the extracellular side of the channel, but then there also has to be the ability of a pore to form that ions can transit through, okay? And it's the ligand binding that actually triggers that and opens the gate so that ions can flow. Okay, so it turns out that there are a lot of plants and animals that actually know how to manipulate channels in other organisms. And we may end a couple minutes early today, but here is the Deathstalker scorpion. I don't recommend being stung by these, but they contain toxins, channel toxins. They contain in the sting and the venom a chlorotoxin, which actually blocks chloride channels. 3 different potassium channel blockers, okay? So these things here are going to affect your nerve function. It's going to probably affect cardiac function, all sorts of things. And that's an interesting thing. But it does give you the idea that, a toxin is a little bit like a drug sometimes, right? Because if you could control such a thing, you could actually develop drugs that would modulate channels, and that might have some therapeutic value. So for instance, you might increase neuronal activity or decrease neuronal activity by using drugs that target channels. You can also, very importantly, change the way that some of the nerve cells that interface with the heart function. And so there are a lot of plant toxins, like digoxin, for instance, that can do these types of things. And so these things will then alter the action potential and the ability of nerves that control, perform in brain function or control the heart rate activity to function. So a lot of actual channel drugs were discovered from natural sources that were originally poisons. And it also means you got to be careful in how you use these drugs because neurotransmission is pretty important. But a lot of the drugs were developed from what were originally toxins, and then the rest is history after that. So we're a couple minutes early, it's okay. But any questions on all this stuff? I hope you got a real feel for how an action potential works. And I also want you to know that the sodium-potassium exchanger pump is especially important, has to generate that voltage difference across membranes. And now you know that voltage, they can open due to voltage and also due to particular ligands. Okay? So next week, or next Wednesday, we're going to actually talk about transporters. And so we're going to talk about channels that transport things other than ions, drugs, for instance. Okay? All right, happy Monday on the final week, you guys. Almost there.